Hello students, I am teacher Svetlana, and in our today's lecture we will learn about acellular organisms like viruses, viroids, and we will also study about lichens, which is covered in the chapter Systematics of Living Organisms. Now, we are studying them separately as these organisms are not included in the 5-kingdom classification system by Whitaker's. So let's begin. Acellular organisms. These are those organisms in whom the body is not separated as different cells. Viruses. Now, the viruses are non-cellular obligate parasites that are characterized by having an inert crystalline structure outside the living cell. So once these viruses infect a cell, they kill the host cell and they take over the machinery of the host cells to replicate themselves. So this name virus, that means venom or poisonous fluid, was given by Pasteur D.J. Ivanovsky. Then we have M.J. Bijovinic after observations that they were able to migrate in agar gel. Agar gel is a nutrient medium which is used to grow bacteria or viruses in artificial medium. Thus demonstrated that the extract of the infected plants of tobacco could cause infection in healthy plants and call the fluid as contagium vivum fluidum, which is called as a contagious or a poisonous fluid. Stanley demonstrated that viruses are inert outside the host cell and can be crystallized. So this is a brief story or description of the history of discovery of viruses. You can have a look at this image here, students. This is how an uninfected tobacco leaf looks. This is how an infected tobacco leaf looks. This is the structure of the virus causing tobacco mosaic virus disease. This is called as an TMV particle. This is how the TMV virus looks. It has a capsid, and within it, it has its RNA, that is its genetic material, that is embedded. M. Stanley showed that viruses could be crystallized, and crystals contain largely of proteins. So these crystals consisted of proteins which were inert outside their specific host cell. Now, in addition to the proteins, viruses also contain genetic material that could be either the RNA or the DNA. Viruses lack their own cell machinery. Now, what is this cell machinery? This is all the requirements for a cell to divide. So they do not have these requirements within their cell. Now, they have protein capsid here. They have protein coat, which is the capsid around their nucleic acid strands, thus considered to be as acellular organisms. Now, viruses are inactive outside their host cells. We know about it that they are inert, they do not have any cellular machinery. Then the question arises, how do they multiply? So once they enter their specific host cell, they take charge of the cellular machinery of the host cell. So the organisms that they infect, they take charge of the host cell and they duplicate themselves. So they utilize their host cell cellular machineries to multiply. Viruses thus can be called as infectious nucleoprotein particles. Here you can have a look at the different types of viruses, their shapes, their structures. Currently, in the current scenario, we are hearing and seeing too much about this individual virus, that is the coronavirus. So let us see some more characteristics of the viruses in detail. Let us have a look at the types of viruses, students. Now, as per the genetic material, viruses are grouped as DNA or RNA viruses. Let us have a look at their structure. Protein coat called capsid is made up of small or smaller units called as the capsinomers. Now these capsinomers is this head-like structure. Now capsinomers are arranged in polyhedral or helical form. So they are either in a helical or a polyhedral form. Capsid protects the genetic material. You can see within the capsid you have the genetic material that is embedded. The genetic material in viruses is either single-stranded RNA or single or double-stranded RNA or double-stranded DNAs. So have a look at this chart. You can see how they are categorized on the basis of their genetic material. This is the enveloped double-stranded DNA. So these organisms have a double-stranded DNA which is enveloped. Then they have these organisms have a double-stranded DNA which is not enveloped. Then you have these organisms which have single-stranded enveloped RNA. This organisms have single-stranded non-enveloped RNA, then this have double-stranded non-enveloped RNA. So this is how they are classified into different types on the basis of their DNA and RNA. Viruses that infect bacterial cells are called as bacteriophages, which normally have double-stranded DNA. So the disease caused by these viruses to a wide range of hosts. Viruses cause disorders like leaf curling, yellowing, mosaic formations, etc. in plants. You have also heard of foot and mouth disease in animals or the swine flu, which are all viral diseases. Then we have viruses causing diseases like mumps, smallpox, herpes, and influenza, along with AIDS in human beings. So on the basis of their host also, they are classified as plant viruses, animal viruses, and the ones that infect the bacteria, which are called as bacteriophages. Then students, let us see what are viroids. Potato spindle tuber disease was found to be caused by single-stranded RNA which lacked a protein coat. Theo Diner in 1971 reported that this is low molecular weight RNA and smaller in size than viruses. These infectious RNA strands are called as viroids. Now these viroids are infectious RNA molecules, and they cause plant disease. They interfere with plant metabolism. So basically they are disease causing organisms to plants. So their host is plants. They are transmitted just the way viruses are transmitted. And then for additional information, we also have prions. Now these prions are infectious protein molecules. Their host is either humans or animals. They cause both animal and human diseases like isomania, mad cow disease. They are very resistant to disinfectants. Now the exact mechanism of theirs is not known, whether there is a change in the shape of the proteins or because the proteins become non-functional. So these are also infectious molecules which are capable of giving disease. Now students, let us have a look at what are lichens. Lichen is coexistence of algae and fungi for mutual benefits. Algal member, the phycobiont as it is called, mostly belongs to cyanobacteria, that is the blue-green algae or green algae. The fungal member is called as the mycobiont. They are excellent example of symbiosis. Symbiosis is the association between two different organisms. Now, functions of the individual members. The algal component of the lichens provides food to the fungal part, while the fungus provides shelter to the alga and also absorbs water and minerals to the alga. The association is intense and it is very difficult to identify them as separate living beings. Have a look at this image here. This is the lichen, the green crust-like structure. Now we have different types of lichens. We have fruticose lichens, which appears like a shrub. Then we have the crustose lichen, encrusting, which is flat and adheres to the surface. Then we have the foliose leaf-like lichens. Now students, let us have a look at the role or the importance of lichen in the environment. Though found in extreme environments like snow-clad poles, lichens are extremely sensitive to pollutions. They are not found in polluted regions, hence are considered as pollution indicators. Lichens also play an important role in soil formation by using specific acid productions. So you can have a look at this image here. These are also called as our air monitors. We have the leafy lichens, we have the bushy lichens. So these act as air monitors. Now, how do our air monitors function? How do lichens tell us whether our air is clear? We have certain categories of lichens that hates air pollution. These lichens are not found where air is polluted. So especially they are extremely nitrogen sensitive. So where there is extreme amounts of air pollution, in those areas these lichens do not grow. Don't like air pollution, nitrogens very much. These lichens are found where there is only a little pollution. So this are found where there is a little pollution is there. Then these lichens which are grouped here, they like some amount of air pollution, little bit of nitrogen, especially the nitrogen from bird poos and fertilizers. So these are very common on our roofs. So this can tolerate pollutions, but we have a category of lichens which are extremely sensitive. So hence lichens are used as ecological indicators. Now brief history about using lichens as bioindicators for air pollutions. You have air pollutants like nitrogen dioxides, heavy metals, O3, HF, other organic pollutants that cause disappearance of lichen from cities and industrial areas. In 1866, it was noted that lichens disappeared from Jardin Limesco near Paris. So smoke from burning of coal was the cause. H2O from burning coals damage these lichens. Hence, lichens have been used as bioindicators of air pollution worldwide. So, it is a different category of organisms, but they play a very important role in our environment. They are natural indicators of whether our area is or our region is polluted or not polluted. Thank you, students, for participating in this lecture. We will meet soon again with the next topic. Till then, take care, be safe, and keep learning.